Okay, so welcome in another video lecture from application in recombinant DNA technology. From today we will going to start our discussion on various kind of transgenic plant. Okay, so now let's start. So first thing that today we will going to discuss is the antisense RNA technology. Okay, and it is generally used to construct transgenic tomato plant. So before going to discuss about how transgenic tomato plant is created, it is necessary to first learn what is the story behind this, why we need transgenic tomato despite we have normal tomato. So the idea is, whenever we laid normal tomato from the tree, there is an enzyme called beta galacturonase, or sorry, not beta galacturonase, it is polygalacturonase, that is the PG enzyme. In short, it is called PG enzyme. So polygalacturonase is activated. This polygalacturonase is nothing but a enzyme. This enzyme causes degradation of various important things inside the cell. Okay, and this degradation facilitates the ripening of the tomato, and as a result, after 2 to 3 hours of it will be totally rotten. Okay, that means, suppose if we late tomato in morning, then by evening it will be totally rotten, right? So that is the normal course of the tomato, and this natural phenomenon, because tomato plant not yield tomato because we can use them for our purpose, but tomato plant produces tomato for its own life cycle, because after tomato will be removed from the, or will be dropped from the tomato plant, it will be rotten, because this rotten tomato will be act as a nutrient for the seed that is in the soil. Okay, so as a result, which will facilitate the further growth of the tomato plant, right? So that is why this rotting process is totally natural. Now, but this natural process drastically affect the industrial and agricultural world of the tomato, and which reflects in the price of the tomato, and as a result tomato become too costlier, and which is normally become unaffordable by normal people like us. So under such circumstances, recombinant DNA technology again come into the picture to protect us from that problem. So here we are going to use the antisense RNA technology. So what is the meaning of the antisense RNA? So I want to discuss with you that the mRNA, because I hope you know that RNA is of 3 types. One is tRNA, another one is rRNA, and the last one is mRNA. And all the 3 RNA participates in the protein forming process actively. That tRNA will help in bringing the amino acid, rRNA will help ribosome to produce the peptide consisting of several amino acid, and mRNA is directly involved, because the codon available on the mRNA will be interacting with the anticodons present in the tRNA, and which facilitate the insertion of the amino acid depending on the what is the codon present, right? So here our main focus will be mRNA. Okay, so as polygalacturonase is a enzyme, and enzyme is protein in nature, and according to central dogma, that simply means that polygalacturonase is coming from some kind of mRNA. Okay, so scientists thought that we cannot prevent the PG from doing its action. Okay, it is not possible. So they thought to do something to this mRNA, which is ultimately facilitating the formation of the PG gene, therefore facilitating the formation of the PG enzyme. Okay, so let's see what they do. So try to understand it simply, that suppose that this single stranded DNA molecule is present inside a cell. Okay, and I have created a complementary single stranded DNA, which is exactly complementary to that red single stranded DNA molecule already present in the cell. Now if I can insert this green single stranded DNA inside the cell somehow, in any means, by any means, so there will be a chance that this green single stranded DNA will undergo a complementary reaction with the already present red single stranded DNA, because they are complementary to each other, and as a result it will facilitate a formation of double stranded DNA molecule, right? That is the basic principle of the nucleic acid hybridization, right? Similarly, the similar thing will be applicable in case of RNA. That means if we can able to form a RNA molecule which will be exactly complementary to the mRNA, which facilitate the translation of the polygalactic uranase, so there will be a chance that it will facilitate a formation of double stranded RNA, right? Now the question is, what is the utility of forming a double stranded RNA? To understand this, you must have some basic knowledge of translation. So remember it very carefully, that a single stranded mRNA molecule will be translated into protein with the help of ribosome. Okay, so translation of single stranded mRNA molecule is possible, but translation of any double stranded RNA is not possible, right? So if we can facilitate the formation of double stranded RNA molecule including the mRNA that facilitates the formation or translation of the PG, that is polygalacturonase, can we inhibit the formation of the polygalacturonase? Because as it cannot translate, it cannot leads to the formation of the PG enzyme, right? So we will going to do this with the help of recombinant DNA technology. So that is what we are doing. Here it is sense mRNA, here it is antisense mRNA. They are together forming a complementary mRNA that facilitates the inhibition of the PG enzyme formation. So that is the idea. So now let me introduce that there is no such process that can help you to transfer a mRNA molecule inside the cell. Up to that part that we have learned in our UG level. Okay, so under such circumstances we have to convert that RNA into single stranded DNA, because we know that normal gene cloning technique is available to transfer a DNA molecule inside cell, right? So we have technique for conversion of RNA into cDNA, which is known as reverse transcriptase. Okay, so this reverse transcriptase will be helping us to convert the mRNA into cDNA molecule. Okay, so now we will converting the single stranded mRNA, which is complementary to the mRNA encoding PG gene. That means, and then we will converting that single stranded cDNA into double stranded DNA. That means what? That means that in reality we are inserting the PG gene responsible for the production of the polygalacturonase. Yes, that is true. We are actually inserting the PG gene, but in opposite orientation. That will facilitate the formation of a antisense mRNA. Okay, that means suppose the DNA sequence GAATTC3' is responsible for the conversion of a mRNA that will facilitate the formation of PG protein. So we will insert CTTAAG. Okay, that means exactly opposite orientation. So that will lead to the formation of RNA which will be exactly complementary to what responsible for the production of the PG enzyme. So there will be a formation of double stranded RNA. So that is the idea. So we will incorporating the double stranded DNA molecule responsible for the production of the mRNA indeed. Okay, that mRNA will facilitate the formation of PG protein. But here we are inserting that double stranded DNA responsible for the production of the mRNA in opposite orientation, so that they can lead to formation of the antisense mRNA. Okay, so now it is clear to us that what kind of DNA we have to insert, what is our gene of interest, it is now clear to us. Now the second important thing that we have to derive, that what kind of technique we should choose. So as it is a story about plant, and we all know that transferring any kind of gene of interest inside the plant, there is one and only very useful method, that is our Agrobacterium tumefaciens mediated gene delivery system. I am referring this as AT. Okay, so here we will also going to use that Agrobacterium tumefaciens mediated gene delivery. So first of all, we will take a typical binary vector. This typical binary vector, let me introduce, will contain everything that a normal TI plasmid have, but it will lack only one thing, and that is important for the transfer of the tDNA, that is the VIR genes. Okay, so this typical binary vector will contain the left border, the right border, and in between left border and right border it will have a cloning site for inserting our gene of interest, and also a NPT2 gene for kanamycin resistant selection, and also it will have another marker gene for selection of bacteria, and bacterial origin of replication it will also have. Okay, so along with gene of interest, as you can see that here is no kind of promoter present, but after insertion of the gene of interest we are willing expression, right? So we should incorporate some kind of promoter that will help us to express the protein PG. So that reason we are incorporating a CAMB strong promoter. Okay, then the fragment from 5' end of the cDNA which will encode the polygalacturonase cloned in antisense orientation. Antisense means opposite orientation. Okay, that means the formation of antisense mRNA. So we will incorporate them and ligate them into cloning site, which will ultimately lead to the formation of the PG antisense plasmid. So here our gene of interest incorporated, this one. Now after that, the next goal will be, we will transfer that plasmid into a E. coli cell via a process called heat shock CaCl2 method, because remember first we will transfer them inside a E. coli cell. Okay, so there is a only method that is super useful for transforming E. coli cell, that is heat shock CaCl2. So we will transfer our plasmid successfully inside E. coli cell. Now after some time we will isolate that PG antisense plasmid from the E. coli cell, and then we will transfer them in a Agrobacterium cell. And now this, remember, this Agrobacterium cell will contain, will already contain a helper plasmid. So why the name helper plasmid? Because remember, this helper plasmid contains a gene, that is the VIR gene. This VIR gene will help that binary plasmid vector, or you can say the PG antisense plasmid vector, to transfer its tDNA portion, that means in that case the gene of interest containing portion, into the plant cell nucleus. Let's see how it is done. So first of all, after the formation of the Agrobacterium tumefaciens containing helper plasmid and our binary plasmid, we will select them with appropriate antibiotic containing medium. Then after that we will grow them in a plate. This plate will contain appropriate antibiotic, obviously, no doubt about this. Along with it will contain acetosyrinzone. This acetosyrinzone is a phenolic compound that that naturally helps the weed genes to be activated. Okay, so this is a broad molecular mechanism that facilitates the activation of the weed gene by acetosyrinzone. We will not going to discuss here about this, but just remember that this acetosyrinzone helps to activate those weed genes. Okay, and as now weed gene is activated, it will start formation of the weed gene proteins, and these weed gene proteins will make nicks at the right border as well as our left border. There will be two kind of weed gene that play here the main role. One is weed D1, another one is weed D2. So these two protein will involve in the nicking process of our right border and left border, which will facilitate the formation of a single stranded tDNA. Now the weed D2 protein will act as a payload protein and guide that single stranded tDNA part, that is here our transgene, into the plant cell and then into the nucleus, and into the nucleus it will be integrated with the plant chromosome, as you can see. So now let's repeat one more time. So idea is, here we are constructing a typical binary vector. What is the difference between normal TI plasmid and binary vector? That normal TI plasmid have a weed gene region, but typical binary vector lacks it. Okay, and it have all the important things except the typical TI plasmid. Okay, and another important thing that typical binary vector lacks, that I have not mentioned earlier, that is, it do not have any kind of opine synthesizing or metabolizing gene, right? And opine is also important for the normal Agrobacterium infection, right? So now we will construct the gene of interest, which will also contain a promoter for production of the mRNA molecule very fast, because we cannot rely on plant for the production, and as a result, if we will rely on plant, then the concentration of the antisense mRNA in the plant cell will be negligible, because it will only replicate when it requires, and which is once during the cell division, right? But it cannot generate too much amount of the antisense mRNA if we only use the plant cell machinery for the transcription of the antisense mRNA. Okay, that is why we are incorporating a personal promoter for the antisense mRNA, that is the CAMB strong promoter. That is how we are constructing the PG antisense plasmid. After that, we will transfer the PG antisense plasmid inside a E. coli cell. Okay, now after some time we will isolate that PG antisense plasmid from the E. coli and we will transfer them in a Agrobacterium, which will already contain a helper plasmid. Helper plasmid means a plasmid which contain the weed genes. That weed genes is not present in that typical binary vector. Okay, and this weed gene will help in the transfer of the tDNA portion of that typical binary vector. That is why the name helper plasmid. Okay, now after that we are having normal chromosome for the Agrobacterium along with a helper plasmid along with the typical binary vector, or you can say PG antisense plasmid. Okay, now we will add, first of all we will select those genomic material containing Agrobacterium, okay, with the help of appropriate antibiotic that we have incorporated. Now we will add or how we will grow those kind of agrobacterium in a plate which will contain acetosyringone. This acetosyringone is a phenolic compound and which will facilitate the activation of the virgins. This virgin will facilitate the production of what? Vir proteins. These vir proteins will involve in making nicks in the red border and the left border, okay, and which will ultimately leads to the formation of a single stranded transgene that will help us, or that will transfer inside the plant cell and then plant nucleus, and it will be guided by two Vir protein named VD2 mainly, and VD1 will involve along with VD2 in the nicking process of left border and right border. So this VD2 will guide that transgene inside the plant cell and it will be integrated with the plant cell nucleus, and now there is the generation of the new tomato plant cell. Now we can grow them to form the callus, and now we can apply them in the soil, which will ultimately lead to the formation of new transgenic plant. That is, we will regenerate the plant and transgenic tomato plant is ready. Now inside that transgenic tomato plant, in every cell inside the transgenic tomato plant, in every cell there will be a constant concentration of antisense mRNA molecule, okay. It will be produced, it will be destroyed, it will be again produced, again destroyed. But whenever there will be a expression of PG gene, that is our mRNA molecule responsible for the production of the polygalacturonase, these antisense mRNA molecule that are present in the cell due to the transgene that we have incorporated will form a double stranded DNA molecule by interacting with the normal mRNA, and as a result that black mRNA molecule that is responsible for the polygalacturonase production will no longer facilitate the polygalacturonase formation, and as a result the tomato will no longer be ripening too fast that it used to do usually, and as a result it will reflect the, or it will affect the price of the tomato, and now the tomato will be easily affordable for us, okay. So that is the entire process of transgenic tomato plant formation. But I want to again told you that does not mean that this antisense mRNA molecule will permanently inhibit the production of polygalacturonase, because if it will happen then it will not facilitate the production of further tomato plant, because I have told you that this rotting process is necessary for the nutrients of the tomato seeds that is present in the soil, but it is just delaying the process so that we can enjoy our tomatoes as a salad. That is the idea, okay. So I hope this video will be helpful to you, and thank you for listening to this class.